In this explainer, we'll be examining a 2025 study from the journal Molecular Neurodegeneration. This research focuses on the midbrain, and it uncovers a potentially critical role it plays in the progression of Alzheimer's disease.
One of the enduring puzzles in Alzheimer's research is its variability. After a diagnosis, some individuals experience a rapid decline, while for others, the disease progresses much more slowly over many years. This study aims to investigate a biological mechanism that might explain this difference, something that could be acting as an accelerator for the disease.
For some time, the scientific community has had a general consensus on the sequence of events in Alzheimer's. It begins with the accumulation of amyloid-β plaques in the brain. This buildup is thought to trigger neuroinflammation, which in turn leads to the formation of tau tangles within neurons. Once this tau pathology emerges, cognitive decline typically accelerates. But there's a problem with this model. The amount of amyloid-β plaque in a person's brain doesn't reliably predict how fast their disease will progress.
To understand that other factor, we need to introduce two key cell types. The first are microglia. You can think of these as the brain's resident immune cells, its first line of defense. They are essential for responding to injury or pathogens. However, when they become chronically activated, this immune response can become harmful, causing widespread, damaging inflammation.
The second cell type are astrocytes. These are support cells that provide structural and nutritional stability to neurons. But in a state of sustained neuroinflammation, astrocytes can become reactive. When this happens, they can stop performing their supportive duties and instead begin to contribute to the ongoing damage.
So this brings us to the midbrain. Researchers had previously observed a compelling clinical correlation. Patients who showed signs of a smaller or less active midbrain tended to move from mild cognitive impairment to full dementia more rapidly than others. This observation provided the starting point for this investigation.
And that observation led to this study's central hypothesis. What if the link wasn't just a correlation? What if the degeneration of specific nuclei in the midbrain, the ones that produce neurotransmitters like dopamine and serotonin, wasn't just another symptom of the disease, but was in fact an active driver of the pathology?
Now, to test that hypothesis and move from correlation to causation, the researchers designed a controlled experiment. They used mouse models to isolate the specific effect of midbrain damage, separate from all the other complex factors at play in Alzheimer's.
Their setup involved two distinct groups of mice. First, they used standard, healthy mice. This group would allow them to observe the effects of midbrain damage on an otherwise healthy brain. The second group consisted of what are known as TG2576 mice. These mice are genetically predisposed to develop amyloid-β pathology, serving as a model for Alzheimer's. This second group was designed to show how midbrain damage might affect a brain that is already vulnerable.
The method they used was highly precise. The researchers induced a lesion in specific nuclei within the midbrain. The goal of this lesion was to disrupt the flow of dopamine and serotonin to the hippocampus, a region of the brain that is central to memory and often one of the first areas affected by Alzheimer's.
Let's first look at the results from the healthy mice. These findings show the direct effect of losing these neurotransmitters without the complicating factor of pre-existing Alzheimer's pathology.
The results were quite clear. The lesion did, as intended, deplete dopamine and serotonin in the hippocampus. This depletion then triggered a significant neuroinflammatory response. Specifically, the microglia became activated through a known inflammatory pathway, the NLRP3 inflammasome. What this demonstrates is that the loss of these neurotransmitters is, by itself, sufficient to cause inflammation. One critical detail emerged. This inflammatory response only occurred with the combined loss of both dopamine and serotonin. Losing only one of them was not enough to trigger this effect.
So that's what happened in a healthy brain. The next and arguably more critical question was what would happen when the same lesion was introduced in the Alzheimer's model mice, those already developing amyloid-β pathology. Here, the comparison between the Alzheimer's mice with the lesion and those without it revealed a substantial difference across all metrics. Microglial reactivity, which was moderate in the control group, became severely exacerbated. Astrocyte reactivity, which was at a baseline level, was robustly induced. Perhaps most importantly, tau pathology, which was absent in the control mice, appeared in the lesion group. Finally, the existing burden of amyloid-β plaques also increased markedly. The midbrain lesion appeared to accelerate the entire disease process,
From this, the study could map out a mechanistic pathway. The loss of dopamine and serotonin initiated the cascade by activating the microglia. These now hyperreactive microglia then in turn activated the astrocytes, and it appears to be these reactive astrocytes that then promote the formation of tau tangles and accelerate the buildup of amyloid plaques. It's a clear chain reaction.
The findings described a significant acceleration of pathology. But the researchers took the study one step further. They asked the next logical question. If losing these chemicals causes this cascade, can we mitigate the damage by replacing them?
To test this, they administered therapeutic drugs to the Alzheimer's model mice that had received the midbrain lesion. They used L-DOPA, a precursor to dopamine, to support the dopaminergic system, and fluoxetine, an SSRI, to support the serotonergic system.
Looking first at astrocyte reactivity, the intervention had a noticeable effect. The untreated mice with the lesion showed high levels of reactive astrocytes. By contrast, the mice treated with either L-DOPA or fluoxetine showed significantly reduced reactivity. It seems that restoring either one of these neurotransmitter systems was enough to quiet this part of the inflammatory response.
And this effect was also seen in the next stage of the pathology. Just as with the astrocytes, the untreated group developed high levels of pathological tau. But again, treatment with either L-DOPA or fluoxetine significantly lowered its formation. This provides a direct link, suggesting that restoring the chemical balance can interrupt this pathological cascade.
So, what are the broader implications of these findings for how we approach Alzheimer's disease? They point toward a new way of thinking about the disease's progression.
The study's authors conclude that their results highlight a pivotal role for midbrain damage in amplifying the neuroinflammatory cascades that drive Alzheimer's pathology. In other words, this damage isn't just a side effect of the disease, it appears to be a central part of the mechanism.
So the primary takeaway is that this research establishes a direct cause and effect link. It connects the health of dopamine and serotonin neurons in the midbrain directly to the neuroinflammation that appears to accelerate the disease. It offers a potential biological explanation for why disease progression can vary so much from one person to another.
This, in turn, points toward the potential for a more targeted, precision medicine approach. It's conceivable that, in the future, a patient in the early stages of cognitive decline could be assessed for midbrain health. If deficits are found, a targeted therapy using dopaminergic or serotonergic agents could begin early, with the goal of slowing that inflammatory cascade before it fully takes hold.
Which leaves us with a final important question. For many years, the primary focus of Alzheimer's research has been on developing ways to clear amyloid and tau from the brain. This research suggests a new and complementary strategy. Could actively preserving the health and function of the midbrain's monoame systems represent a new frontier in how we prevent, slow, and ultimately treat Alzheimer's disease?